I am going to turn things over to Alana. Alana will be moderating today. She is the Associate Director for NICO. We're so thrilled that she's joined the NICO team. But in addition to being Associate Director, she is also the Senior Director of Scientific Partnerships for Kellogg's Center for Science of Science and Innovation, or CSSI. So Alana, thank you so much for being here today, and I'm going to turn things over to you.
Thank you, Megan. Thanks so much, and thanks to everyone for joining. I will echo Megan's sentiment that it's been a great series, and we appreciate everyone showing up for these dynamic lightning talks. We have 4 speakers from a variety of disciplines and topics. We will kick off the conversation with Jaehyuk Park. Jaehyuk is a postdoctoral fellow at Kellogg and NICO, working with Haejin Yoon. And Jaehyuk made the extra effort to go into the office today because, interesting fact, his cat Mochi was a little bit vocal today. So we look forward to a cat-free presentation from Jay. Thank you so much.
Yeah, thank you for the great presentation. And this is Mochi for the reference. And yeah, I'm Jaehyuk Park, and postdoctoral fellow at Kellogg School of Management and NICO. And today I'm going to talk about people, place, and ties, which is about landscape of social places and their social network. Let me start from one incident in New York. So January 2014, there's a conflict between senior Korean citizens in New York and the management at McDonald's at Queens, New York, because these old citizens actually ordered a very cheap and basic food like French fries and coffees and stay a long time, every day with their friends to hang out with their friends. So it becomes kind of serious about political issues and social issues. And at the end, actually, there's another New York Times article coming out that the journalists try to find out why that McDonald's. And the journalists actually find out that there's a citizen center in Flushing, New York, where this happened. But the journalists found out that actually because of the accessibility and price and independence, these citizens are more likely to use McDonald's, which is nearby their resident area, than senior citizen centers. And this is not that weird situation in the United States these days. So you can find some of the other articles that especially fast food restaurant becomes kind of social places in our era, especially for minorities and the poor people. Since these social places have been known as a really a critical in our social life and city planning, based on a lot of good work by Robert Putnam and Jane Jacobs, and also especially this third book by Ray Oldenburg introduced a new concept about third places, which is different from the first place, which is our home, and second place is where our workplace, and the third place is representing the places that we are meeting our friends and acquaintances and make a new social relationship. And this concept of third place becomes kind of critical in multiple areas like a city planning and public policy, like a Brookings report, and also businesses. So Starbucks announced themselves as a kind of third place. However, existing studies on third places are really focusing on very limited in specific region, for example, Cambridge in Massachusetts, and they surveyed the resident or city dwellers in that area to ask what kind of places are considered as a social places for you, and etc. And there's no kind of systematic nationwide study about the landscape of third places. So here, with collaborating with the core data science team at Facebook, this study provided geographical and demographic landscape of third places in the United States using Facebook pages. So this is a kind of example of NICO's Facebook pages, so you can see what face pages mean. As a first, we start to check the representability of Facebook pages. To do that, we found that actually 3 categories, restaurants and places for worship and bars and pubs, are appeared in social places, and at the same time, they're appeared in county business patterns, US Census data set as a kind of business categories. That means it allows us to compare that the number of Facebook pages in a US county versus actually establishments measured by census. And as you can see, there's a reasonable correlation, at least in log scale, between the number of businesses and number of Facebook pages in the similar categories. And then we check the geographical landscape of different types of third places. And this is a distribution of 6 example social places. So as you can see here, places of worship is hardly more concentrated on the south area of the US, while bars and pubs are more concentrated in the Midwest. And as a person who have lived in Midwest during the last at least 7 years, I can't agree more with these bars and pubs. And also you can see restaurants and community amenities seems kind of complementary to each other within a kind of similar neighborhood. And I can give you 5 to 10 seconds so you can find some interest patterns by yourself so we can discuss at the end of this lightning talk, I think. All right. And then we also checked how demographic characteristic of a region is related to the emergence of specific third place categories. So the figure A shows that how the urban and rural area have a different distribution of third places. And as you can see, place of worships and community amenities are more in rural areas. And some of business-oriented social places, which is like a beauty shop and restaurants and rental places, they are more overrepresented in kind of suburban areas. And finally, place of worship has a negative correlation with the income of a region as well as education and foreign-born populations. After that, we check the social network of fans for each pages. So first here we extract a representative fan network for each third place category. So we first randomly sample 25,000 Facebook pages per each category and their social networks. And those pages are having followers between 50 to 50,000, which allows us to exclude some extreme pages. And then we measure 18 different topological statistics, network statistics of those all the sampled business pages. And then we extract the closest network having the most similar statistics with the average statistics of each category. So as you can see, different types of social places have a kind of little different types of social network and fan network. So here, maybe the prevalence of dyads and triads, such as like outdoor and indoor recreation and parks and monument here, or restaurant here, that represent people are more likely to visit those kind of places with already their small group of their already known friends, rather than the customers that already know each other all together. And also, if there's some core and periphery structure, you can find small group of people are very know each other, while other people are just weakly connected of one of those core people. We can imagine that those kind of places, the people, there are some regulars who likely to visit almost very regularly, every week or every day, while other peoples are randomly visit and they are kind of have a friendship with one of those core members. And places for worship and bars and pubs and community amenities, those places have this kind of characteristics. And finally, using this 18 topological network measurement, for each pair of social place categories, for example, performing arts and bars and pubs, we created a random forest classifier model, machine learning model, to check that which cluster or which types of categories are hard to distinguish by the machine. And if, so that means if you see here, the score gets closer to 0.5, that means it's really hard for random forest classifier to distinguish those places, which means their kind of social network structure are similar to each other. In contrast, if like some of the numbers are very like close to 1, that means it's easily distinguishable that two social places, third place categories that end up mediating different types of social network. And we can find some of the clusters here, like clubs and societies, indoor recreation and performing arts, bars and pubs, and community amenity, those 5 clusters. And 2, retail and large sport venue has more likely to have like similar types of social network, while those 3, non-urban outdoor recreation and restaurant and parks and monument, are likely to have kind of similar types of social network. And interestingly, place of worship has a very unique social network structure compared to others in our analysis. So take-home message is we generate a nationwide landscape of social places across their types, and we compare the embedded social networks of those places to check if different types of social places are distinguishable by the social relationships they mediate. And hopefully we can share the data set about like, county level, business type, and number of Facebook pages and their social network structure for the future studies of certain places. And currently we are working on this with a legal team on Facebook. So these are my collaborators, YY and Indiana University, and Mike Daly, Boston State, and Monica Ball at Facebook. So this is my presentation, and thanks for your time and concentration. And if you have any question beyond our Q&A session, just don't hesitate to contact me through any of this, my contacts. Thank you.
Great. Thank you. Mm-hmm. Thank you very much. Lots of interesting information packed into that talk, Jay. Thank you very much. Next up, I would like to introduce Emma Zajdela, who is a third-year PhD candidate in engineering sciences and math, applied math, working with Professor Danny Abrams. She is an NSF Fellow and a 2020 Buffett Institute Impacts Fellow. Couple of fun facts about Emma. She is a classically trained violinist, as well as a yellow belt in judo. So we look forward to a powerful presentation, Emma.
Hi, everyone. I'm so thrilled to be here today. I love Wednesdays at NICO's, not just for the delicious lunch that we get each week, but for the food for thought. And so today I'm excited to share with you a taste of my own research, which is about catalyzing collaborations and the dynamics of team formation at conferences. So first I wanted to start a little bit with how I got interested in this question. When I was an undergrad, I worked on game theory research on poker, and I became really fascinated with this idea that we can use math to understand human behavior. And at the same time, I was working with a nonprofit called the Malta Conferences Foundation that uses science diplomacy as a bridge to peace in the Middle East. And working on these 2 projects, I began to wonder, can we use mathematical modeling to help science diplomacy in regions of conflict be more successful and understand why it is successful in the first place? So then I moved to Northwestern and started working with Professor Abrams on the physics of social systems. There's many questions that could come up from this, one of which is, what is the role of intervention by organizations like the Malta conferences, and are scientists who attend these conferences more likely to form new collaborations and cross-border collaborations? But the question we're going to focus on today is a little bit more general in terms of how effective are conferences at forming new collaborations. As researchers, we spend a lot of time at conferences, and it's important to know that these conferences, which cost a lot, not just in terms of money, but in terms of time investment, time away from research, are at forming collaborations between scientists. So we developed a mathematical model that predicts which people who co-attend a conference are likely to form a new collaboration. And more specifically, we look at the probability that a pair of participants at a conference will collaborate, given their previous knowledge of each other before the conference and also their level of interaction throughout the conference. In 1996, the sociophysicist Zajdela said, "Do humans behave like atoms?" So when we set out to develop this model, we thought about scientists like molecules in a solution. And when molecules are together in a solution, like scientists at a conference, their probability of forming a reaction rises with increasing time spent interacting. More intense interactions or a higher concentration will make the probability rise faster. And then when the interaction between the participants ceases, the probability of the collaboration forming will decay. So this was our initial model. And as you can see, it's a dynamical system with a strengthening term corresponding to the first part and a weakening term when the scientists stop interacting. But there was a problem with this model, which is that after a while, after a certain period of time, the probability that the scientists would collaborate relaxed to 0. And that would imply that people completely forgot each other after attending a conference, which isn't realistic because when you meet someone at a conference, maybe you wait a year and send an email and then you develop a collaboration with them a year later. So the next model that we developed is a nonlinear model that incorporates this assumption that if people have interacted sufficiently, they will remember each other. And so their probability of collaborating with each other will remain above a 0 level. So the linear model could be described as resulting from a potential. And by formulating it in this potential way, we could improve it to make it incorporate the elements of the nonlinear model. So in this first figure, you can see this is when there is no interaction between participants. And there are 2 fixed points in this potential, one at 0. And by fixed points, you can think of it almost like a ball rolling down a series of hills and valleys. So the lowest points would be the fixed points. So in this case, the probabilities fixed points would be at 0 of the people collaborating, and then a higher level, which corresponds to that memory probability. And as participants interact throughout the conference itself acts as a catalyst, like a catalyst in a chemical reaction, increasing the reaction, and it does so by deforming the shape of the potential. So as S, which is the strength of interaction between the participants, increases, the potential deforms and the equilibrium points change. So you could imagine, again, this ball rolling down a hill, except this time it's reaching a different equilibrium. And it's, for example, in the fourth figure, would reach the equilibrium of 0.3. So the probability of the individuals collaborating would be this higher level. This is an example of how the model would perform. So in the top figure, this is the interaction between 2 participants, so a pair of participants at a conference from the time of the start of the conference, 0, to the time of the end of the conference. And you see there's a peak at about 3 or 4, and this corresponds to these 2 individuals attending a session together. And then there's another peak a little bit later, and that corresponds to those 2 participants attending a second session together that was more a smaller session, so the interaction was more intense, which is why you see that value being higher. And on the bottom figure, this is the solution that the model gives with these parameter values. So you can see that at the beginning, the probability of the scientists collaborating rises, and then it rises again as there's a second peak, the second interaction later. And the value at the end of the graph at time t equals almost 30 is the value of the probability that these people will collaborate. So if you're interested, I actually made this into a Shiny app, so you could visit this website and try out different values of the parameters for yourself and see how that would affect the value of the participants collaborating. So now let me talk a little bit about the data that we have for this model. So we partnered with the Scialog conferences organized by the Research Corporation for Science Advancement. And we analyzed 12 conferences out of 4 series of conferences over 5 years. That corresponds to about 13,000 pairs of potential collaborators. The structure of these conferences is a little bit different than some other conferences. It's by invitation only, about 50 fellows per conference, and a very interactive format, which is aimed at generating new collaborations. So the Research Corporation for Science Advancement collected pre-conference survey data, which means we know exactly how well each person who attended the conference knew each other person before attending the conference. And throughout the conference, we also know which sessions each of the individuals assigned to together, which is how we were able to construct the timeline for each pair of participants of what that interaction looks like. And at the end of the conference, the fellows submit proposals, some of which are funded, some of which are not. So for the purpose of this research, that's what we were understanding collaboration as meaning, was submitting a proposal together. Now, what does this look like? So this is an example for one conference, and you can see there's about 1,300 pairs of participants at this conference. And I zoomed in on the pairs of participants who ended up submitting proposals, which you can see is sort of a small fraction of the total pairs of participants. And each horizontal stripe corresponds to the interaction between one pair. So if you look, for example, at pair number 4, you can see that this pair interacted at one session, then another session, and then no sessions towards the end of the conference. And the darker the color, the more intense the interaction was. Also, the background color would correspond to the initial knowledge of each other. We tested what the effective interaction time at the conference was for the groups of people who didn't interact versus the people who did interact. And we found that groups who collaborated together spent a lot more time than groups who didn't interact, who didn't collaborate, sorry. And in this example of one of the conferences, groups who collaborated spent on average 41% more effective face-to-face time than those who didn't collaborate. And that's about equivalent to being in a group with 12 people for an extra hour. We performed this analysis over all of the conferences and also looking at different aggregations of the conferences. And you can see that with extremely high statistical significance, the people who collaborated had a higher initial knowledge of each other than the people who did not, and also had much stronger interaction than the people who did not. However, if we looked at the proposals that were awarded versus denied, we didn't see anything significant between the fact that people who submitted proposals and were awarded or people who submitted proposals and were denied. Finally, our current work is focusing on testing the model and comparing it to some other models, and so far the results are promising. The future work, we're also planning on testing this model over several years, not just at one conference, by combining it with conference data and publication data, so to see exactly what the timeline of 2 participants would be over a period of several years. And this work could also help us think about optimal conference design. How, given a certain conference and certain parameters, could we optimize it to try to form as many collaborations as possible? Finally, we're also interested in going back to the question of the Middle East and seeing how collaborations that form at conferences in a region where it's more complicated for these conferences to be formed, these collaborations to be formed. Thank you so much to NICO. Thank you to all my supporters. And please feel free to follow me on Twitter or email me if you'd like to talk. Thank you.
Thank you very much, Emma. Really fascinating talk. It'll make us all think a little bit more closely about our connections when we're at conferences. Next, I would very much like to introduce Gary Nave. Gary is a postdoctoral fellow in the Department of Engineering, Science, and Applied Math, also working with Professor Danny Abrams. Gary joined us in September. Interesting fact, in spite of that, he has not set foot once in Evanston because of the pandemic. So we will very much look forward to welcoming Gary to Evanston, and please join me in welcoming him to his talk currently.
Perfect, thank you. Yeah, so I have yet to visit Evanston. I've been to Chicago twice, but pandemics are weird, and I'm still living in Boulder, Colorado, which is where I did my first postdoc. So today I'm going to be talking about a topic from my PhD work at Virginia Tech. It's a long and complicated title, but I'll hopefully get to each piece of it as we go. This is kind of more of a method-y talk than a specific application. But yeah, I hope it's interesting to a lot of people. So I'm interested generally in flows, and the first question might be, what do I mean by flows? You probably initially think of something like this. So this is the wind as of about 8:30 Central Time last night in the US. So there's kind of high-speed wind flowing up toward the Midwest, of course. There's lots of swirly wind over here near where I am in Colorado. And so it's natural to think of flows of air or water, but we can also think of flows in terms of models. So this is the kind of Van der Pol oscillator. This is just kind of a simple 2D mathematical model. Or my first postdoc, I was actually working with groups of honeybees, and I'm interested in kind of the flows of collective animals moving near each other. So honeybees use kind of their motion on the honeycomb to communicate with one another. So flows could mean kind of a huge variety of things, but anywhere where you have kind of lots of potential states, and those states are changing over time. So for those who have taken like a differential equations course, the first step when you think about solving when one thinks about solving a differential equation is thinking about kind of finding the answer. And so what you usually think of in terms of an answer in a first differential equations course is given an initial condition x naught, how does that evolve over time along your flow? So you're thinking about a single trajectory starting at a single initial point evolving over time. But you can also take kind of a more global view of the flow and think about if you just have a blob of initial conditions and evaluate that blob for a given time, t, then how does that blob deform? And I don't know why I drew straight lines on the top and bottom as my blob deforms, but yeah, you could think about how maybe all of the initial conditions evolve over a given time. And there's a type of feature in the flows, or really kind of a lot of features in a class of features in flows that I would refer to as coherent structures. This is kind of the typical picture. So these are hyperbolic coherent structures. The red one is a repelling curve, and the blue one is an attracting curve, and the green blob is a set of initial conditions. And over some time tau here, that green blob moves away from the repelling curve and is attracted to the attracting curve. So you kind of, your circular blob becomes this oval along the attracting curve. And these types of features are really interesting. They provide kind of a skeleton of the flow. I've pulled a couple examples from interesting papers. So somebody looked at coherent structures in ocean flows. I believe this is the Eastern Atlantic Ocean near Africa. And there's all these kind of swirly coherent structures here. And they mapped where they observed seabirds fishing. And so it's kind of hard to see, but there's a bunch of blue dots and green dots here near these coherent structures and some red dots near this one. And so they were able to show that seabirds forage along coherent structures. And this is related to kind of as time evolves, material kind of moves along these coherent structures. So they provide some organization to the really complicated flow. There's also a paper last year proposed search and rescue along attracting structures. So they called these traps. I forget what the acronym stands for, but the idea is that there's this attracting feature in a flow, and if there's kind of a man overboard situation, then people can look at models of ocean flow and try to predict where those people are rather than just doing kind of a grid search along a vast ocean. One method for kind of one particular type of coherent structure is the trajectory normal repulsion rate. And so if you think about a dynamical system where your state is changing with some vector field v, and then we think about the time t flow map where we're mapping all of our initial conditions to their conditions after time t, then you can think about tangent and normal vectors to each initial condition. And how, for this metric, we look at how the normal vector changes over our integration time t. So this is our picture. So our initial normal vector here and naught is mapped to this kind of second red arrow and with the gradient of the flow map. And then we look at kind of the normal component of that. So what we're looking at is the normal growth of the initial normal vector. And so here's kind of an expression for the trajectory normal repulsion rate. And we were interested in kind of what happens when we look at the infinitesimal normal growth. So the biggest, or a big barrier to calculating these is it takes a long time to integrate a lot of trajectories on a computer of your model or ocean flow or what have you. And so if you have a trajectory here and a nearby trajectory up here, then you look at that mapping of the normal vector, and we look at what happens as our time gets closer and closer together, and then look at the growth rate at an infinitesimal time of this normal growth. I'll kind of skip over the math, but we arrive at something that we call the trajectory divergence rate, which relates to normal vectors and the Eulerian rate of strain tensor. And we're looking at instantaneously, are normal vectors growing or shrinking, and by how much? So looking at an example, we can cook up the simplest thing I can think of that has these interesting features is x dot is x. So things are growing horizontally, and y dot is negative y. So things are shrinking vertically towards the middle. And when we apply the trajectory divergence rate, then we end up with this interesting structure. If there's weird curves, it's how the computer renders it sometimes. It's actually a nice smooth gradient. But the most positive trajectory divergence rate is here along the y-axis. And that's where trajectories are repelling from one another. They're getting further apart. And the most negative here is the x-axis, where trajectories are coming together. So then we can come up with this simple schematic. So as we calculate this thing, the trajectory divergence rate, negative values indicate attracting curves and congregating trajectories. Positive values indicate spreading apart or diverging trajectories. It's useful. There's no need to calculate the flow map. It's computationally inexpensive, and it shows both repelling and attracting structures at once. And you don't need to pick an integration time. You can just kind of figure out what is the instantaneous growth rate. One scenario where this works particularly well is in thinking about slow fast systems. So this is a fun textbook example. I can't imagine this actually happening in real life, but we have a hula hoop with a bead on it that can slide along the hula hoop, and then we spin the hula hoop on its end. And when we do that, there's the math works out to this kind of nice system that relates the two rotations. And if there's a lot of friction on our hoop, then all of the action, all the interesting stuff in the system happens along this curve. And when we apply the trajectory divergence rate to this, we're able to pretty accurately identify what that interesting curve is. And we show that it's strongly attracting, and yeah, there it is. Another class of systems that might be interesting is limit cycles. This, you could think of kind of neuron activity, you could think of heart rate. This is just kind of a simplified mathematical limit cycle, but there's a lot of really interesting potential applications. You could think of walking. And as we calculate the trajectory divergence rate along this kind of attracting curve that all the trajectories end up on, we find that here along these slow regions, it's very strongly attracting. But there are a few regions along this limit cycle where trajectories are actually getting further apart. And so if you have, say, an arrhythmia and a heart rate, and you want to kind of move to a different cycle that the heart can do, then maybe these regions where trajectories are getting further apart is a good place to push it. So if you were able to think about what's the best place to shock a heart, for instance, to knock it into a better heart rhythm, then these unstable regions may be really interesting places to apply that. And then one more picture, we can look at the ocean flow off the coast of Cape Cod to show that this is useful for ocean flows as well. You can calculate kind of Lagrangian coherent structures here in red and blue, and those tend to appear in the same regions that the trajectory divergence rate indicates that there's a lot of convergence or divergence. Yeah, thank you very much for the time. I didn't put my contact info on here, but you can find me on Twitter as well at GKNave. And it's really fun to chat with you all here at NICO.
Thanks very much, Gary. Interesting research, and your example of using this method to potentially predict flows and of ocean flows and where a person overboard might be. That's really valuable, tangible application of this very interesting research. So thanks for that example.
Yeah, no problem. Thank you.
Next, we would like to welcome Sarah Ben Maamar. Sarah is a postdoctoral research associate in the Department of Chemical and Biological Engineering.
Sorry about that.
Oh,
sorry. I'm just sharing my screen again. It's just easier like that.
I'll let you go ahead and take care of that, Sarah, and just finish up for a moment here, describing your expertise. And we're very thankful for your leading role in organizing the Data Science Nights. A fun fact about Sarah is that she is French and she has a passion for visiting national parks. She has visited 15 parks within the last 2 years, and looks like we are all set up with the presentation. So welcome, Sarah.
Thank you very much for the nice introduction, and hi everybody. It's a pleasure to present this work today. So today I'm going to present a work that has been actually initiated by Sophia Liu, who was a PhD student in the Emerald Lab and who left now. And right now, Reese Richardson, which is a new PhD in the lab, and myself are contributing to expand this great project. So today, I'm gonna present about the reproducibility of computational tools used in RNAseq bioinformatic pipelines. And I'm gonna start with this, basically this slide, just to show this study that has been performed by Baker and published in Nature in 2016. And basically in this paper, they asked about 1,500 researchers if they thought that there was a reproducibility crisis in their field. And you can see immediately that about 90% of them actually responded, yes, there is definitely a crisis in reproducibility in my field. Then they have been asked also, particularly in their own field, if they have personally failed to reproduce any experiment, either their own or if somebody else experiment. You can immediately see that in all the disciplines, researchers failed to reproduce results. And chemistry and biological fields are particularly affected by these issues. So in biology, we use a lot right now what we call next generation sequencing technologies. And the reason is because this technology provides, really produces a lot of data for very cheap. You can see here, for example, the cost of sequencing that has drastically dropped since the 2001 up to 2008 and stayed pretty stable until pretty much now. And this obviously led to a huge increase in the utilization of this next generation sequencing technology in biological studies. And the increase has been steady up to now. So obviously, I cannot talk about biology without setting the stage with this central dogma about biology. So in biology, we assume that DNA molecules are present in all the cells that constitute a body, an organism. And this DNA molecule gets transcribed into RNA molecules, then get translated into amino acid sequences that end up basically being your proteins. So from the same DNA molecules, you may know or not that you can actually produce different cells, different cell phenotypes with exactly the same molecule. And how is that possible? It's just because we have this step that is called transcriptional regulation. And it is basically allowing the cell to regulate which gene it wants to express and which gene it doesn't want to express. So example, in this blue cell here, you can see that the gene 1 is expressed, but this gene, the same gene 1 is not expressed in these orange cells. And this is how we get two different cell phenotypes. So when we look at the gene expression, we actually look at transcriptome profiles here framed in red. And those transcriptome profiles, are obtained by RNA sequencing. What is RNA sequencing? So very briefly and in a very simplified schematic way, in order to do RNA sequencing, you need first to extract all the messenger RNA molecules that are present in a cell. So this is what you can see here. You have different molecules that are coming from different genes. You have a red gene, and you have 5 molecules of this red gene here, 3 molecules here of this orange gene, and 2 molecules of this yellow gene. Once you get those molecules, you fragment them, and then you sequence them. And from the sequencer, you get this type of raw data that basically is a bunch of sequences made of ATGC. From there, you cannot say much as a biologist. So you need to go through this computational pipeline. And the first step of this computational pipeline is basically to quality control this raw data. So that means basically removing part of those sequences, especially at the end of the sequences, because usually they are of lower quality and we want to work with only high quality data. Once you have done that, you can continue with the alignment step. And the alignment step is basically to map back all your clean sequences to your reference genome. So here you have a portion of this reference genome that is made of a red gene, an orange gene, and a yellow gene. And you can see that we map those fragments that we produced before to those different genes. Following this step, you then end up basically producing your profile, your transcriptome profile. And it's basically through the counting step where you just, basically it's very simple. You report just the number of fragments that are mapping each of your genes. So for example, if you look at the gene 1 here in red, it has the highest expression here because 5 fragments are mapping it compared to 4 fragments for the orange gene and 3 fragments for the yellow gene. All right, so for each of the steps, here's the issue. We have multiple algorithms that are available. So first of all, from the raw data, some scientists perform this quality control step that I mentioned before, but some other just don't. For the alignment step, we have multiple algorithms available. I just listed 7 of them here. Same for the counting step and same for the differential gene analysis step. So if you just look at those different algorithms, you already have like 84 possible variation just by mixing up the different softwares you can use in each step. But you add to that the fact that some of them have different modes and some modes have also their own parameters. You also sometimes researchers uses 3 different reference genome to map their reads. And you also have like about, 3 parameters for each of those algorithms. So at the end of the day, you end up with about 54,000, more than 54,000 variation of pipelines. So basically, if you don't document how you analyze your data, it's almost impossible to retrieve, to understand how data has been analyzed in each paper. So what we started to do basically, and I don't want to spend too much time on this aspect, I'm happy to go more into detail if you have any questions, but we first look at the documentation, how people are reporting what they are doing in terms of RNA-seq sequencing. And so we looked at the PubMed central database, and we did a bunch of steps to filter out some specific papers because we wanted to focus on only papers that are focusing on Escherichia coli, which is a model organism in microbiology, and that are using RNA-seq approaches. So after all of our filtering, we ended up with a data set of about 75 papers here that are actually really dealing with RNA-seq on Escherichia coli. And so we compared this final data set. We looked basically at the distribution of this data set in terms of number of papers between 2013 and 2017. It's just a time frame we decided arbitrarily. And we compared that to basically all the other papers that have been published in the same period just to see if our data set is representative of what happens generally in the biological field. And you can see it on the yellow line is pretty much the case. So from this experiment, from this first filtering and data set, we then, since previous studies have reported that about 50% of the studies are reproducible, we then basically draw this line, red dashed line that you can see here, which represent this 50% of reproducibility. So from our 75 papers here on the y-axis, we expect that 50% of them should be reproducible. And we looked at this reproducibility in terms of documentation for each of the steps, and this is what we got. So you can immediately see that, first of all, not a lot of steps are reproducible. And actually, almost, besides the raw data, the availability of raw data, almost none of the steps are reproducible. And even worse, for the quality control step, which corresponds to the trimming and filtering step here, sometimes it's not even performed. So we already see immediately from this graph that we don't have a lot of reproducibility. Then what we did is that basically, if you look at those 75 papers and you remove each of the papers for each step that are not responding to your reproducibility criteria in terms of documentation, we see that at the end of the day, we have we have 0 publication that actually is fully reproducible in terms of documentation. So what we show here already is that we don't have any methods of reproducibility because we don't have enough information to repeat the experiment in our data set. We also looked at the different version of pipelines that are existing in the literature. And so we basically looked at, if you start with the same raw data, can you obtain the same results at the end if you use different pipeline? And this is just a sample of what we did, obviously. But here you can see on the Y, this is a heat map that shows the correlations in results between different counting algorithms. You have feature count, cufflinks here, HTSeq here. And so if you have like a red color, that means it's a Spearman correlation calculation. So that means that basically the results are highly correlated. They are in agreement. On the contrary, if it's blue, that means that the results are not in agreement. And you can see, for example, if you compare HTSeq with in its mode B and C, and you compare it to its mode A, in general, you have a good correlation in the results. So it's pretty much coherent. But if you look at HTSeq in mode B and C, and you compare it to cufflink, you still have an agreement in the results, but it starts to be much more blurry in terms of correlation here. But worse, if you look at the feature count and HTSeq in the mode B or C, you can see that here the results are completely in opposite direction, which is basically showing that we don't have either results reproducibility because the results that we produce depending on the pipeline we use are not the same. And finally, I want to show here just the results of the gene expression for different pipeline variation that we performed. So for example, here you can see that overall, all the pipelines that we tested, different versions, 65 of the genes that we found were detected as non-differential expressed, which is totally fine. From the genes that are differential expressed that are shown here, you can see that only 5% were found positively expressed in all the different pipelines we used. 10% of them were found negatively expressed in all the pipelines we use. But worrisomely and interestingly, a lot of these differentially expressed genes were found in some pipeline positively expressed, in some other pipeline negatively expressed. So that shows that basically here we have no inferential reproducibility either because the results that we're putting are not the same and we cannot draw the same conclusion because our results are different. So in conclusion, I just want to emphasize here and the results of this study in general is that we lack methods, results, and inferential reproducibility for computation pipeline using RNAseq, and we absolutely need a need for computational standards on how RNAseq data should be processed and analyzed. And with all that, I thank you and happy to take any question.
Thank you very much, Sarah. Very interesting work. And I'd like to open it up for questions. We have a couple in the pipeline. We'll kind of start in reverse order. Jay, you had a question from your first talk on a very specific one. Why did you, and this is from one particular slide, though I can't recall which one, why did you have about 0.5 AUC in the diagonal? I'm sure you know which slide this one was. He said the person asking the question says, I would expect good predictability there. Thank you.
Yeah, I think this will be the slide 7. And yeah, I'm glad that you enjoyed the talk. And so basically this model is the differentiation between two social place categories. So if they are really similar to each other, that it becomes near 5 or 0.5, which means like random chance. But if it's totally distinguishable each other, that it becomes like close to 1. So like random forest classifier can differentiate or distinguish those two social place categories or network from two categories very well. So that's why like almost diagonals are like 0.5 or 0.49, which means they're basically same. So like it's hard to differentiate or, yeah, that means, is that a good, yeah.
Thanks.
Mhm.
Thank you, Jay. And I lost the person's name who asked that question, so I can't tell you who it is if they have follow-up questions. But the next one we have is for Emma from Thomas Whitehock. Thomas asked, do you have information, Emma, about the researchers who attended the conferences, for example, fields of PhD received, the institution from which they received it, and can you say anything about how these variables may have influenced collaborations?
Really interesting question. We do have a lot of information about the researchers who attended. This information and then more information about their demographics, for example. So it's a question we get often, and I think it's probable that there is maybe a homophily factor, for example, or between different researchers. So people who are more similar tend to associate more, and that might be true for collaborations. But the reason that we didn't look at these is because we wanted to see if beyond these factors, maybe demographic factors or field of PhD, et cetera, if just interacting more would lead to more collaborations.
And Emma, for the data sets that you used for those interactions, and I apologize if I missed that at some point in your presentation, but how did you measure physical interactions?
Yeah, that's another good question. So basically the scientists attend sessions together, and there's mini sessions and large sessions. That's the two type of sessions that are. One is about 12 people, one's about 4 people. And so the way we did it is that we have exactly who was in which session. And then we assumed that the larger the session, the less important the interaction was. So a mini session with 4 people would be worth more than a larger session with 12 people.
So, something that may be interesting as follow-up, you may be familiar when we hosted the International Conference on Computational Social Science in 2018, one of the NICO postdocs, Wouter Vermeer, he had badges, physical badges, had a chip that would calculate the closeness of participants in that conference. He's now a faculty at Feinberg, so that may be an interesting piece of work to look at if they still have that data.
Yeah, that is, I've heard about that data before. That is something that would be really interesting to kind of capture also the more informal interaction that people might have in between sessions.
As we're waiting for more questions to roll in, I might ask one of Sarah. And Sarah, could you expand a little bit upon the applications of your research?
Thank you for the question. I'd say the applications are pretty broad, but what I think the main attention we're trying to get is to make scientists, particularly in biology, aware of the impact of their choices in terms of how the way they analyze data, and to maybe become more and more critical about the choices they are making when they decide to analyze their data in some way or another. I think in the field, unfortunately, or fortunately, I don't know, a lot of scientists tend to just basically mimic what has been done before without necessarily like this critical thinking about, okay, if I do that, what do I infer implicitly or explicitly? And so this is, I think, the main, I think one of the the biggest takeaway of this work and also impact. And yeah, we are really trying to expand, like disseminate the message and convince biologists that we can all do, because I'm including myself in the pack, of course, that we can all do a better job in the way we analyze our data.
It's important consideration. Gary, may I ask you the same question about application? I know you had a couple of them in your presentations, but I'm sure there may be a few others that hit the cutting room floor. If you wouldn't mind expanding on that a little bit, that would be interesting.
Yeah, there's one more in particular that I thought was really interesting. I thought about including it, but I didn't go hunting for the news article. I just happened upon a little story, like a 1-paragraph story in a publication somewhere, about some research that somebody's done during COVID, about how if you roll down the passenger window and the opposite rear window in a car, then air will flow kind of diagonally across the car and act as a barrier between the person in the driver's seat and the person diagonally in the back seat. And so they're using that as kind of a active filter of air. So I guess one thing I didn't go into is that a lot of these geometric structures act as barriers to transport. So these highly stretching regions of a flow don't allow particles to pass through. So there's been research on plant pathogens in the atmosphere, and you can track these structures and see where those plant pathogens will spread to, even in neighboring states. And so there's been a little bit of work, I know, and I don't know that if they connected it to coherent structures or not, about how these kind of high velocity flows can act as barriers to COVID transmission as well. Yeah. It's also my favorite intuitive explanation is when you walk into a big box store like a Sam's Club or a Costco or something, they have a fan blowing straight down by the doors, and that prevents the cold or warm air from passing as easily through the barrier of the doors, and therefore saves on the heating bill. So.
Excellent. Those are very relevant application in today's world. So thank you, Gary, for sharing that.
Thank you.
Mm-hmm. And I think we have covered most of our questions, and we are at 1 o'clock. So we covered a lot of interesting topics, right? This is what's so interesting about NICO, the cross-disciplinarity and the interesting breadth of topics that we cover, from collaborations in the physical world, physical spaces and conferences, flow features, and looking at Sarah's talk on reproducibility, all very interesting topics. Thank you all much. And Megan, did you have any closing comments?
You pretty much covered it, Elena. Thank you so much for moderating today and for fostering discussion. And thank you again to Emma, Gary, Jaehyuk, and Sarah for your presentations. They were so interesting. And we will see everyone again when our spring quarter starts. So be on the lookout for that communication from us. All right.